So I'm really excited, like a kid on Christmas Eve, because we're finally at the end of this story. After this lecture, we will understand exactly what happened to the glucose that we consumed, like 6 lectures ago? And as we move past this material, we'll simply talk about how excess glucose is stored and what happens to excess pyruvate, and switch gears completely away from glucose metabolism. So this is it. This is wrapping up this big multi-lecture story that we've been telling for quite some time. The name of the process we'll be talking about today is oxidative phosphorylation, and this is the second half of chapter 20, and the first of only 2 lecture chunks to cover this material. In this lecture chunk, I just want to make sure that we've gotten the take-home concepts from the last electron transport chain lecture straight in our minds and refresh our memories as to where we're picking up from. And we'll talk a little bit about how we link what occurred in the electron transport chain to OXPHOS, which is the affectionate abbreviation of oxidative phosphorylation. The next chunk will talk about the process itself. It'll talk about the mechanism of oxidative phosphorylation, how we make ATP using a proton gradient, and we'll wrap that chunk up by talking about the overall energetic yields of the complete metabolism of glucose. And by energy yields, of course, we mean ATP yields. So this essentially is the electron transport chain as we described it in the last lecture. We had electrons arrive at the ETC on NADH plus H, the reduced form of NADH. We called that the NAD shuttle. And NADH plus H brought those electrons to complex 1, offloaded them to FMN, and then they were passed to iron-sulfur clusters. This was where the first set of protons were pumped, because FMN could accept those protons, whereas the iron-sulfur clusters could not. And eventually the iron-sulfur clusters passed those electrons on to CoQ. CoQ was a mobile shuttle. It traveled through the mitochondrial inner membrane, and it brought those electrons and protons to complex 3. Complex 3 was this strange case where we can only transfer 1 electron at a time. So CoQH2 passed 1 electron on to complex 3, and then another CoQH2 had to pass its electron on, and then CoQHs passed electrons and protons within each other. We talked about that quite a bit in the lecture. But eventually, these electrons make their way through complex 3, and cytochrome C is reduced as a result. Cytochrome C carries those electrons on to complex 4, where they're passed on to a bunch of different cytochromes within complex 4, eventually those electrons making their way onto molecular oxygen. Oxygen is reduced to water, absorbing protons from the matrix, further contributing to the gradient, and we're at the end of the line for those electrons. You might notice what's not shown on this diagram is complex 2. Complex 2 is also known as succinate dehydrogenase. It sits in the membrane. It is holding electrons and protons on its covalently bound FADH2. Those electrons and protons make their way directly to the CoQ shuttle, and CoQ brings those electrons and protons to complex 3 as I've just described. So always keep in mind that complex 2 is kind of out of the mainstream ETC here. It converges on complex 3 through CoQ as well. So once we're done with the electron transport chain, we've offloaded our low-energy electrons onto oxygen, making water, but much more importantly, we've stored all of the energy of those redox reactions throughout this chain as a proton gradient. We ended that last lecture with a severe proton gradient, lots of protons in the inner mitochondrial space, very few protons in the matrix, and the inner mitochondrial membrane in the way, stopping them from flowing. And this is the diagram that I showed at the end of that last lecture to show that, and I made the point that essentially we're going to treat this like a dam, where the inner mitochondrial membrane is the dam itself. We've got a lot of water or protons on one side of that dam, and much less water or protons on the other, and of course water always wants to flow from where there's more of it to where there's less, and these protons are no exception. So we have that energy stored as a proton gradient, where protons are literally dying to flow across the inner mitochondrial membrane, but they can't because that membrane is severely semi-permeable, and without a channel, those protons have no way to move. So the question becomes, what do we do now? Well, somehow we have to take this idea, this proton gradient, and use it to make ATP, and that's what oxidative phosphorylation is all about. So to make a single ATP molecule from ADP to power the phosphorylation of ADP into ATP, we need about 30.5 kilojoules per mole. And once we put that energy into ATP. That's the energy we get out when we break that last phosphate off of the triphosphate chain. So each ATP molecule stores about 30.5 kilojoules per mole because that's what it took to make that molecule. So that's the energy we need to make ATP. And when we have all this potential energy stored as a proton gradient, we have to consider something else, though. Those protons are all positively charged. And that positive charge, the effect of that positive charge can move across the membrane. So what kind of ion do you think is attracted to that positive charge? What kind of ion do you think is lining up here on the inner surface, on the matrix surface of the inner mitochondrial membrane? Opposites attract, so those are anions. Anions are lining up there, and this is creating an even more attractive force. So if we thought those protons wanted to get into the matrix badly before, just based on their concentration gradient, how do you think they feel now when they're also being drawn electrostatically to these anions? This is what we call an electrochemical gradient. And so these protons really, really want to flow. How are we going to link that potential energy to making ATP? We need something that will allow that potential energy of the proton gradient to be released into kinetic energy and simultaneously trap that kinetic energy and use it to make ATP. Said more simply, we need a way to couple the physical flow of protons across the inner mitochondrial membrane to ATP synthesis. And believe it or not, a single protein complex is that link. This protein complex is called ATP synthase, and that name just tells us that this is an enzyme that makes ATP without using ATP, which makes sense. And this is what ATP synthase looks like. Probably, in my own opinion, one of the most amazing proteins in all of nature. And I hope by the end of this lecture you'll agree with me. Again, based on the name of this enzyme, of course it makes ATP, and it's ATP synthase that allows the energy stored up in that proton gradient to be released and converted into making ATP. ATP synthase is made up of two large separate domains. The F0 domain, which spans the membrane itself. The F0 domain spans across the inner mitochondrial membrane here. And the F1 region, which is also called the head group, or the head region of ATP synthase. This is where we'll actually make ATP. And I'll give one more bit away here. Somewhat obviously to some of you, perhaps, since this is the transmembrane region of ATP synthase, the F0 region, this is going to be our proton channel. The protons are going to flow through the membrane by flowing through this F0 region. And again, we'll be making ATP up here in the F1 head. Now, if you don't believe me, here's an electron micrograph of the inner mitochondrial membrane. What you see indicated by this top arrow is the membrane itself, so we'll trace the membrane here. And each of these regions studying that membrane are the F1 regions of ATP synthase. So amazingly, we can visualize these proteins with our own, I almost said naked eye, not with our naked eye because we need an electron microscope to do it, but we can see these proteins. They're that big. It's extremely rare to be able to directly visualize any protein. Only a few have been visualized, but this is one of them that we can actually see with the aid of an electron microscope. So schematically, this is what we're looking at in that electron micrograph. We're looking at a fragment of the inner mitochondrial membrane, and each of these kind of bulbous, I don't know what you'd call them, buds, are the F1 head groups of ATP synthase. So here, each of these red circles attached to these sticks is also the F1 head group of ATP synthase. It's pretty beautiful. The electron transport chain and ATP synthase itself are a coupled process. In fact, ATP synthase goes by another name, which is complex 5, to show that it comes directly after complex 4 of the ETC. But we can uncouple these processes as well. Excuse me there. And this uncoupling can be achieved by some toxic compounds. I'm saying obviously toxic here. The reason why I hope it's obvious is because if we uncouple ATP synthase from the electron transport chain, we're essentially leaving the mitochondria with no way to use the proton gradient to make ATP. This is the cell's primary source of ATP, and so the cell very quickly runs out of ATP and dies as a result. So it is toxic to have ATP synthase not working. When this uncoupling is achieved, The electron transport chain still works. We still reduce oxygen into water. We still create a proton gradient, but we can't release the potential energy of that proton gradient in order to make ATP, and so cells die of ATP starvation. Without these toxic uncoupling agents, we make that proton gradient, we reduce oxygen to water, but the proton gradient is then allowed to flow directly through ATP synthase, and as a result ATP is made, and the cell has plenty of energy to do what it wants to do, and the cell survives. So setting the stage here in this brief first chunk for ATP synthase, hopefully you have a sense of where we're heading next, introduced to ATP synthase itself and tying it a bit to the electron transport chain. We started by reminding ourselves that we ended the electron transport chain with energy stored as a proton gradient, and we had to somehow release that potential energy as kinetic energy and make ATP with it. The next chunk will be dedicated almost exclusively to the mechanism of ATP production, one of my favorite stories to tell in all the courses that I teach, and I hope that you'll be just as amazed with it as I am. Until then, thanks for watching.